In the last one, we talked about the structure of DNA. Now we're going to talk about the packaging of DNA. Now, first I want to talk about why DNA packaging is important. Why can't we just let it float around and interact with everything? Well, that would end catastrophically, that's why. Everything can't interact with DNA. There should be a regulation to how and why and when certain sequences are read and expressed. There's also the problem of DNA damage. If you let it float around, it's going to, well, collide into things, be exposed to radiation, be exposed to aggressive proteins, reactive elements. So it's in everybody's favor to keep it compact in a smaller area. Now, this is especially important for eukaryotes because when you're a bacteria, you have a circular little chromosome and everything's good. However, when you're a eukaryote like me, and you likely, if you have a massive DNA like eukaryotes do, you need to compartmentalize it. You need to have places where you can access DNA easier, and you need to have archives of DNA that you don't usually access. And you can have the archives of DNA where you don't access and use that DNA in another type of cell where it's in a more accessible compartment. So the compartmentalization is not a part of the code itself, but how the code is packaged and how the code is expressed. So this is very important. Let's look into it. First, starting from the prokaryotes, because they're little cute bacteria boys. They keep their DNA in a small compartment called a nucleoid, and they keep it with minimal amount of proteins near their plasma membrane usually. That's about that. Now let's look at eukaryotes. Eukaryotes use a lot of mechanisms to package DNA, but supercoiling is a very important one that I mentioned before, so I want to emphasize that. We use topoisomerases, enzymes that help with supercoiling, to induce or reduce supercoiling depending on how compact we want our DNA. Topoisomerase 1 breaks one strand of DNA, rotates the other strand, and then sticks the strand again, releasing or inducing tension. The same goes for topoisomerase 2, except it cuts two strands, holds it onto them, rotates them, and then sticks them back together. Like I said before, they can both induce and relax supercoils, depending on the demand. And how much something is supercoiled or how much something is interacting with a histone protein or another packaging protein is important, because how much the major groove is exposed to the cytoplasm depends on the packaging. And when the major groove is exposed, as you can see here, anything just can come and interact with it, which means we can read it, we can express it easily. Let's look at how eukaryotes usually package their DNA. There are different stages of packaging, but all of them depend on this protein we call histone, mostly. So let's look at the histone protein. It's a bead-shaped protein that consists of many subunits. We're going to look at H2A, H2B, H3, and H4 first. As you can see, this is H4 and another H4 subunit, creating like a slice type of shape. And four of these slices, the H4 slice, the H3 slice, H2B, and H2A slices, create a bead together. DNA is wrapped around this bead, approximately 2.5 circles, and then H1 subunit comes on top of this bead and fastens the DNA with the bead. H1 sequence also helps this structure form. I'm going to get to that. And since these all have two copies and H1 has one copy, it's found less, exactly half of the amount of the other histones, and binds to the outside. Eukaryotes packaging the DNA can be seen in many different stages. The naked DNA stage, as you can see here, is basically what you've always seen when you thought of DNA. But it's never the case within the cell. As you can see, anything could just come and interact with this and disrupt this. So we need to wrap it around something to keep it fastened. That something is the histone. And this beads on a string structure is 11 nanometers thick, which you don't need to know, but it's just a neat fact. This beads in a string formation then becomes more compact and creates this chromatin fiber structure, which is how DNA is naturally found. DNA is never naked or beads on a string. It's chromatin fiber. And this structure is facilitated by the H1 histone, as I've said before. And this structure then creates loops. Loops are also associated with other proteins that help create the base of the loops. I'm going to get to that. And these loops create the larger loops, which create the chromosome segments, which creates the chromosome. And a chromosome is 1400 nanometers thick. Meanwhile, naked DNA is only 2 nanometers thick. Think about that. Histones are basic proteins, which help DNA interact with the histone, because DNA is literally deoxyribonucleic acid, and histone is a base, and acids love bases. Okay, cells regulate DNA packaging using chromatin packaging altering, which means they alter the histones, they alter the proteins that package the DNA by adding methyl groups, acetyl groups, or phosphates to tag the proteins and call other proteins, either modify them, or this chemical modification, this addition of methyls or acetyls or whatever, can chemically induce a more compact structure. And this is done at the tail of the histone proteins. So yeah, they kind of have a little tail where metal can be attached. Keep that in mind. Chromatin also contains a lot of non-histone proteins, one of which is cohesin, which forms the loops, these loop formations in mammals. Transcriptionally active DNA is less tightly packaged, therefore more accessible to outside interaction. And we're going to get to that more in the euchromatin, heterochromatin part.